Hey Beaklings, it's Mr. Boylan, and today I want to talk to you about meiosis, or the creation of haploid gametes. So basically, what's going on here? Well, you have to understand mitosis first. So in mitosis, you started off with a somatic body cell, right? And it's a diploid, meaning you have two sets of chromosomes, one from mom, one from dad. You had the somatic cell, and basically during M phase, you went ahead and ripped the nucleus apart, right? Exactly right. And then you went through cytokinesis as well, to create two genetically identical daughter cells, okay, similar or identical to the parent that you started with, right? It's asexual reproduction. Well, what we're going to do today is we're going to talk about meiosis. So hopefully you've already drawn the pictures. Hopefully you have your head wrapped around meiosis a little bit. And what we're going to do is we're going to take a look at, okay, starting with a somatic cell again, but this time it's in a sex organ, okay? And guys and girls do this a little differently, okay? So let's just talk about a guy today. So here's a somatic cell inside of the testes that's going to go through meiosis. And what that means is, okay, meiosis 1, we rip apart the nucleus a little differently this time, right? And then we go through cytokinesis. We just created two haploid cells, okay, in meiosis 1, because each of these are going to have half the amount of DNA that the parental cell had. They're not gametes yet, though, okay? They're not gametes because they have a little bit too much DNA. So now we have to go through meiosis 2, which is kind of like mitosis with half as much genetic material, meaning each of these meiosis 1 cells, let's jump right into meiosis 2. We do not want to go back into interphase. We want to go right into meiosis 2. So each of these two cells is now going to split again, and that's going to create 4 total cells, and these 4 cells are going to be genetically dissimilar haploid gametes, right? So they're only going to have one set of chromosomes in each of them, and in our case 23 chromosomes, and that can be used for sexual reproduction. Okay, and hopefully you understand a little bit about why sexual reproduction leads to such variation in a population. Okay, if not, go ahead and watch the drawing video first, and the rest of the chapter gets into that too. Okay, so here we go. Let's take a look at meiosis. Okay, so meiosis reduces the chromosome number from diploid to haploid. All right. So meiosis, like mitosis, is preceded by interphase. And that makes sense, right? If you didn't go through S phase, you wouldn't have enough DNA to put inside of all of your cells. And if you forget what S phase is, don't forget that's when you copy your DNA, and DNA replication occurs then, right? And the reason that you have to copy your DNA, don't forget, is so, oh yeah, we don't just want one piece of chromatin, right, a spaghetti noodle. We have to copy that so we have two, and eventually those two pieces can coil up in prophase, in this case prophase I, and they can form sister chromatids, right? So it's very important to go through interphase before you do meiosis. The parent cell divides twice, producing 4 daughter cells, right? In mitosis, we only divided once. And then the pairing of homologs, or homologous chromosomes, which is called synapsis, starts meiosis 1. That's going to be super important, okay? And it's a huge difference between meiosis and mitosis. These chromosomes may exchange corresponding segments, which we know is called crossing over, and that's a huge facet to our genetic diversity as a species. It's basically shuffling your genes. Meiosis 1 produces 2 daughter cells with a set of chromosomes each. That's why I say that those are haploid cells, right? But they're not gametes yet. Just because they're haploid doesn't mean that we can use those for reproduction. Okay, and it's around this time that girls kind of shut down their meiosis preparations, because girls already started making their eggs before they were even born, right? And they shut down meiosis and they finish it later before ovulation. Okay, whereas guys, we make new fresh sperm all day, every day. So meiosis 2 is similar to mitosis, as sister chromatids will separate, right? So that's going to have the genetic material then in a different way. Rather than pulling apart pairs of chromosomes, we're actually separating at the centromere, okay? And that should remind you of anaphase of mitosis, right? And then finally, meiosis 2 produces 4 haploid gametes. Okay, so let's take a look at some pictures and go through the videos. Okay, so here we go into meiosis 1. Well, before we get there, let's actually look at an interphase cell. So this is probably a late G2 cell that's getting ready to begin, in this case, meiosis. And what do we know about a G2 cell? Well, we have centrosomes in there, okay, and they could have already copied themselves. So centrosomes, don't forget, have those centriole pairs, and those are those microtubule organizing centers, right? We also have our nucleus, and we know that inside of here the DNA has already been copied. So technically, we have 92 pieces of chromatin in there, 92 huge spaghetti noodles, or something like that. This cell's probably also getting pretty large, right, depending on what cell type it is, and that means its surface area to volume ratio is probably stressed right now as well. But this is a little different. We're not dividing here to save space. We're dividing here to create gametes, right? Eventually, we're going to do gametogenesis. So let's take a look here and see a G2 cell in interphase.
Like mitosis, meiosis is preceded by an interphase, during which the chromosomes replicate. The centrosome also duplicates in preparation for cell division.
Okay, so notice they're not showing all the other organelles again. We're just moving right along here, but that's a typical cell that's about to start, not mitosis this time, or M phase, but meiosis. Okay, so here we go. All right, in meiosis 1, we start with possibly the most important phase, and that's prophase I. Okay, so let's take a look at what's going on here with prophase I. Well, first of all, huge difference. When you start to coil your chromosomes, this is called synapsis because homologous pairs line up together. So in this case, we have 46 chromosomes in our cell, 23 pair. This cell right here, they're only showing 3 pair or 6 total chromosomes. So in this case, this would be a 2n equals 6 cell, and we're after n equals 3 gametes. Okay, so if you look here, not only does synapsis happen, okay, and this is called a tetrad because it's 4 sister chromatids lined up together, but crossing over also happens in there, and that's a way to shuffle up your chromosomes. So technically, when you're all said and done, you might have created sperm or eggs that have combinations of genes and alleles that have never existed before on planet Earth, right? Because if you think about it, one of these chromosomes came from your mom, and one of these chromosomes came from your dad. So you're basically shuffling your mom and dad's DNA to give to your kid, which is pretty wild stuff here. All right, so let's take a look at prophase I.
Prophase I begins with condensation of the chromosomes. Homologous chromosomes, each made up of 2 sister chromatids, come together in pairs. This pairing is called synapsis. Each chromosome pair is called a tetrad, a complex of 4 chromatids. Chromatids of homologous chromosomes cross over each other and exchange parts at chiasmata, singular chiasma. Meanwhile, other cellular components prepare for the division of the nucleus. The centrosomes move away from each other and spindle microtubules form between them. The nuclear envelope and nucleoli disperse. Finally, spindle microtubules capture the kinetochores that form on the chromosomes, and the chromosomes begin moving to the metaphase plate.
So you notice that we don't talk about pro-metaphase of meiosis, right? And that's because prophase kind of includes that already. So this is later prophase now. So when we did mitosis, I said the hallmark of prophase is when chromosomes coil. When you can start to see those chromosomes, boom, you know that you're in prophase. Well, similar here, only there's a big difference in meiosis. In meiosis, we have synapsis, right? And that pairing of homologs or homologous chromosomes is hugely important, all right? Now, they talked about crossing over, so let's take a closer look at that and what crossing over is all about. Okay. Breakneck speed here, this laptop. Here we go.
During prophase I of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
Okay, so crossing over really is important for our genetic variation as a species, right? So when you have a kid, it's not like you're either giving your kid your dad's chromosome number 2 or your mom's chromosome number 2. No, you very well might be giving your kid a mix-up of both of those chromosomes, right? Which is pretty cool. And if you understand this crossing over as you take future biology courses and we get into the complexities of linked genes and recombination frequencies and all that kind of stuff, you really have to understand crossing over to do those problems later. All right. So here we go. Let's look. What do you think comes after Prophase I is, of course, Metaphase I, right? Now, Metaphase I, it's very simple to understand. It's just the lining up of homologs down the center of the cell, right? So if this was mitosis, you would only see one chromosome in a line here, okay? Or all the chromosomes in a single line, I should say. But in Metaphase I, you have the pairs lined up, and that's really, really, really important. Now, these are fully coiled here. See over here, they weren't fully coiled, but by the end of prophase, they're pretty tightly wound up. So these are fully coiled, okay? So we don't have to worry about the crossing over and all that stuff anymore once we get to Metaphase I. All right, and how did they get to the center? It's the same thing as mitosis, right? The microtubules grab the kinetochores and move them down to this imaginary plate. So let's quickly take a look at Metaphase I of meiosis. There's something else super important that also happens here in Metaphase I. And again, if you already drew the chromosomes with me and drew meiosis out in my other video, you know that independent assortment happens here. And when we talk about genetics later, if you can go back and remember, wow, Metaphase I is how we lined up all of our chromosomes, and that's really important, the amount of different ways that we can line them up. That'll help you out a lot. So Metaphase I, it seems simple, but it's a hugely important step for genetic variability. Okay, let's take a look here. And I lost it. I don't know what I did. Let's try and refresh. Okay. Maybe I could tell some jokes while we're waiting. I never used to like facial hair, but then it grew on me. All right, you're welcome.
At Metaphase I, the chromosome tetrads are aligned on the metaphase plate. For each tetrad, kinetochore microtubules from one pole of the cell are attached to one homologous chromosome, while kinetochore microtubules from the other pole of the cell are attached to the other chromosome of the pair. Thus, the homologous chromosomes are poised to move to opposite poles of the cell.
Okay, so nothing really earth-shattering there, except how you line them up. You have no control over that. So in other words, what if you put mom up here on the top and dad on the bottom or whatever? Yeah, that makes a difference if you did it that way or a different way. Eventually your sex cells are going to be dictated what's inside of there based on how you line them up in Metaphase I. So it's a pretty big deal. Okay, and you know what comes next probably, right? And that's going to be Anaphase I. Now, huge difference between this and mitosis, because in mitosis at this point you all know that, oh, we were going to go ahead and separate our double-stranded chromosome at the centromere. We were going to separate this into sister chromatids, now become daughter chromosomes, right? One chromosome, two chromosomes. Not the case here in anaphase, and that's why we're able to make haploids in meiosis I. All right? What you're doing over here is you're actually separating the homologous pairs from each other. So technically, in this cell, you still only have 6 chromosomes. If we would have separated these at the centromere, we would have doubled the number of chromosomes, but we're at 6 still. So let's take a look at anaphase and make sure that we understand that. Here we go.
During anaphase I, each pair of chromosomes is pulled apart, and the homologous chromosomes move toward opposite poles. Note that sister chromatids remain attached at their centromeres and move as a unit toward the same pole.
Okay, so like we said, very important to understand, you did not separate chromatids at the centromere. All you did there was take chromosome number 1 from my dad and move it this way, and chromosome number 1 from my mom and use it this way, or move it this way. So you separated pairs or homologs there, but you did not separate the sister chromatids. Okay, so now we know telophase I is probably coming next, right? And this is pretty easy, only look where we're at now. We started with a cell with 6 chromosomes, but now we're going to be down to a cell that has 3 in each, right? So once cytokinesis takes over here and splits this into 2 daughter cells, each of those cells is going to be haploid because instead of having 6, we're going to have 3. However, as you can see, they're going to be double-stranded, so we'll have to fix that in meiosis too. Okay, so let's take a look at telophase I and see how we end up with these haploids, even though they're not true gametes yet.
The chromosomes finish their journey during telophase I cytokinesis occurs, producing two haploid daughter cells. Note that each chromosome still consists of two sister chromatids. Meiosis isn't over yet. Remember that it consists of two consecutive divisions. During meiosis II, the sister chromatids will be separated.
Okay, so there we go. We did create haploids. Now in this picture it's different because this is only, we started with 4 chromosomes and now we're down to 2 in each cell. But point is, we did it. We got through meiosis I. We started with, in humans, 46 chromosomes, and now we're down to 23 in each cell. The problem is those 23 chromosomes are still double-stranded. I can't give this to my kid, right? I can only give my kid 1 copy of every gene. So that's what meiosis II is all about. So we definitely do not want to go back into interphase here, right? We're done with the cell cycle. We're going right back into meiosis II. Okay, so the chromatin is going to start to uncoil and things like that, sure, in these 2 new daughter cells, these haploids, but we're going to jump right into prophase II here. Okay, so let's have a look. So, meiosis II, prophase II. Hey, look at this. The nuclear envelope's breaking down. The chromosomes are starting to coil up again. The chromatin is folding into these chromosomes. The mitotic spindle is starting to form again. Wow, it looks just like prophase of mitosis. So what's the only difference? Well, you have half as much DNA because now you have 2 cells doing it. All right, so prophase II of meiosis II, easy stuff. Okay, it's basically prophase of mitosis. All right, let's take a look.
The second division of meiosis, meiosis II, is essentially the same as mitosis. The important thing to remember is that meiosis II begins with a haploid cell. During prophase II, a spindle forms in each cell, and the chromosomes move toward the middle of each cell.
All right, pretty simple stuff, right? And you know what's going to come next. Let's go ahead and line them up in metaphase II, right? So here we go. Metaphase II, it's probably going to look just like mitosis. Yep, not a difference here. Now, if this was metaphase I, we'd have twice as many chromosomes here, and they'd be lined up in pairs, right? Homologs. But you know that in metaphase II, we're down to a haploid cell already. So sure, we're just going to go ahead and line them up down the center so we can split them in half. Let's take a look.
During metaphase II, the chromosomes align on the metaphase plate.
There it is. Easy stuff, right? Okay, here we go. You know what comes next now. Anaphase II, right? And an anaphase II, oh, okay. Now this one's a little bit interesting. Go ahead and think about what we started with in this scenario. We started with 6 chromosomes, and after meiosis I, we went down to 3 in each cell. But technically, now that we're in anaphase II, how many are we back to? Well, we had 3. We're back to 6 again. Because don't forget, this is 1 chromosome made out of 2 sister chromatids. But when you rip them apart at the centromere, we actually have 2 chromosomes here. Okay, so this cell right here actually has 6 chromosomes again. But that's okay because we're going right back through telophase and cytokinesis to make haploid gametes. So let's take a look here. Okay, and click this up.
Sister chromatids are pulled apart in anaphase II.
Okay. Earth-shattering there. So again, and look at the non-kinetic or microtubules, they're still extending and cytokinesis has begun. And finally, we have these 2 cells that are now going to go through telophase II, right? And in telophase II, cleavage furrow is going to form again. That's that actin, that ring of microfilaments is going to pinch off. And now we have 1, 2, 3, 1, 2, 3. Oh, we're only going to have 3 chromosomes in each cell. And now the fact that they're not 3 of these, but they're 3 of these means those we can use for sexual reproduction. Okay, so let's take a look at telophase II and finish this process up. Okay, this is almost to the end of making 4 genetically dissimilar haploid gametes.
In telophase II, nuclei form at opposite poles of each dividing cell, and cytokinesis splits the cells apart. Meiosis has produced 4 haploid cells, each with 1 set of chromosomes.
Okay, so that's pretty much it. Now we have our 4 haploid gametes, and in the videos, again, we start with 4, so it makes sense that all of them have 2, right? So going back to our PowerPoint here, you can see that, oh yeah, all of those cells would have 3 chromosomes because we started with 6. So let's think about a human here. In a human, we would have started with what? 46 chromosomes, and then by the end of Meiosis I, we went down to 23, right? And now where are we at? Well, in Meiosis II, we have 23, which we eventually ripped apart in Anaphase II to create, well, that would be back to 46, but now in Telophase II, they split, so now we have 23 again, right? And that's how you give your kids 23 chromosomes, okay? Now, this is not necessarily gametes here. We have to go through gametogenesis yet, and then that basically means just specializing these cells to make them actually sperm in this case, okay? So that is an overview of meiosis, okay? I hope that made sense. We're not jumping back into the cell cycle here. These sex cells are just sitting there, these gametes waiting to be used, and if ever fertilization does happen, boom, you're right back into a somatic cell going through G1S, G2M over and over and over again, right? So gametes, haploid, all different from each other, very important. And again, what's really important here, and the rest of your chapter, we'll talk more about this, is there were two things that we talked about today that really increased genetic diversity. Okay, the first one was crossing over in Prophase I, hugely important step. And the second one was independent assortment or orientation of those chromosomes in Metaphase I. Right? There's lots of different ways for you to set up your 23 pair of chromosomes, around 8 million actual ways, right, where you could set them up. So that's a huge difference because how you set them up dictates what's going to be in your gamete. And think about this, it takes two to reproduce, right? Because we're sexually reproducing organisms. So the other person that you're going to have produce offspring with, right, they also had to do that. So that's random fertilization. That means you have about a 1 in 8 million shot of your sperm or egg being a certain cell, and the other person has about a 1 in 8 million shot. So when you're going to put them together, that's about a 1 in 64 trillion chance of that offspring being, very specific offspring, right? Or two people. What are the odds that they could have two identical kids? Not talking about identical twins. That's different. But what are the odds that the same two people could have two kids and that they could be exactly the same as each other? And if you think it's about 1 in 64 trillion, not really, because we forgot about crossing over. Crossing over messes that all up too, right? So there's a lot of diversity here that most people, when you talk about why our species is so healthy, they'll understand if they're into biology that, well, we're sexually reproducing. That's a big deal. So besides just the random mutations that can happen throughout your life, we have other ways too that we're very different from each other, and that's really important. Okay, it's a key concept in biology. So I hope you found this video helpful, and good luck studying. Okay, take care.